of chemicals and cells, cell portions, there still is a number of points we can make about muscles and how they work. So we're going to start there. In everyday working life, with this sarcomere action, this actin and myosin cross-bridging, producing contractions, what does that really mean about the body and how it moves? Well, it kind of depends. What job are you assigning to a muscle? A muscle that propels the thigh through walking and running is handling a heavy load, and it's also a muscle where your bodily function works better if you can move that muscle quickly. So that's basically for running. That's a very heavy, quick muscle contraction. Whereas there are other parts that are moved by muscles that are very tiny. For example, blinking your eyelids. That's something that has to happen as quickly as possible. You want that to happen instantaneously because the eyelid over the eye blocks your sense of vision. So you want to swab that eyeball and get it back out of the way so that it doesn't interrupt that sense of vision. That's a heavy, quick load. So we basically think about what's the load the muscle is moving and how long do we expect it to contract when we call upon it. Now, in addition to that load and duration, it's kind of the position of the body, the sustained effort. So there are some activities that go on constantly. They have to be kind of part of the background, like blinking our eyes, beating our heart. There are others that occur sporadically, but when they occur, they basically may need to be sustained right up to the point of exhaustion. So how do we handle this high, heavy load or extended frequency and duration beyond what we'd call our aerobic maximum? I want to point out that range. At rest, like we are now, our muscles are refreshed, loaded, and ready to go. They may be suffering some fatigue from the normal everyday wear, sitting too long in one position like I'm doing now. But it's certainly not true that I'm at the verge of muscular exhaustion based on what I've done today. Let's take a look and try to analyze different ways of approaching this. Here are three muscles. That eyelid muscle illustrates a quick, high tension for the sarcomeres and the myofibrils involved, but basically it's such a light load, it doesn't take many. To blink that eyelid quickly, the tension builds quickly and then releases quickly, so that within between, let's say 3 or 4 milliseconds to 15 milliseconds, barely over a tenth, over 10 milliseconds, we have the completion of that contraction. Now, the gastrocnemius is a muscle, it's your calf muscle, the back of your lower leg, and it basically is the muscle. If you contract in the anatomical position, contract the gastrocnemius as a pair on left and right side, you push yourself up on tiptoes. It's what extends the ankle. It's also active every time you take a step. You might roll forward on your foot, but we gain that extra 6 or 7 inches of stride by contracting the gastrocnemius as our right power step completes. That pushes us forward, and we can reach forward with our left. As we do that, as we're taking a step, we are basically balancing on one foot for a long time. As we swing our left leg, we're basically rolling over our right leg, and balance becomes important. Right to left movement is possible. There's a muscle on the side of the leg that is constantly able to correct the orientation of the calf beneath the femur, keeping the bones upright. Think about what happens. Your foot hits on uneven ground, and it might throw you to the right or throw you to the left, or you might step in a hole. You have to adjust that postural position to make that step a smooth step that keeps moving straight forward. So this is something you have to manage completely through the landing of the foot all the way up to the completion and the lifting of the foot. That's what the soleus muscle does. It's doing less propelling forward and more balancing you on that upright peg, that kind of individual leg. Looking at an individual muscle contraction, it's hard to describe how it happens, but we call it a twitch when we go through one cycle and measure the tension generated. Now notice something. This 0 point is where we apply the neuromuscular stimulus. And there's a delay before any tension develops. Now what's going on during that delay? Neuromuscular stimulus, neurotransmitter dump, stimulation of the sarcolemma, the racing of that electrical impulse down the T-tubules, the release of calcium, the unlocking of the actin-active sites, and then finally the beginning of cross-bridge formation, which then accelerates the tension. But you'll notice this kind of 2.5, 3 millisecond delay, the latent period, while all of those chemical steps are performed. Then as those cross bridges form a maximum tension, the contraction phase, and then you release the stimulus. Those cross bridges basically are forming fewer and fewer as you complete your cycle by 40 milliseconds, which is 4/100ths of a second. That twitch is done. This is what happens in the gastrocnemius muscle, and it illustrates the way an individual twitch is generated within a myofibril. We can use these myofibrils and fibers together in different ways, and we kind of understand this. There are those everyday activities that we can sustain as long as we want to. There are those that tax us, are doable, but they tax us more and more to maximum output, and then there are those that exhaust our muscles and cause us to break down. These patterns are shown on a twitch basis. You'll notice this pattern never reaches the maximum tension in trepi, and the characteristic of these twitches is that they build, but between twitches you're allowing it to return all the way to base, all the way to 0 before you twitch again. So since you're starting from 0 every time, this is a refreshed and fueled muscle, and you can maintain this low level pattern called trepi indefinitely. If you, however, compress these stimuli together so that you're returning toward relaxation, but then you stimulate again, and you build another twitch from that level, and another from that level. You notice the increasing tension that's being built here by the more frequent stimulation. This is called wave summation, and this is how you build greater and greater tension and force in your muscles as you attempt to move more and more weight. But still you're within this range established by the dotted line. This is maximum tension, or tetanus, that can be developed within this system, and you're still in this middle range. When you release the stimulus, relaxation descends all the way to 0 tension. Wave summation can be maxed out at any level between here and here. With our skeletal muscles, we often have to do work. Imagine you pick something up and you're required to hold it. Maybe you're carrying boxes of books in from the truck and they're heavy. So here is a wave summation pattern. As you pick up that box of books, and then you're going to sustain it at this level right here. Not allowed to get any higher than that, but you're burning a lot more ATP up here as the stimulus frequency increases. You reach a kind of a plateau, but since it's well below the maximum force you can generate, you can sustain this for a long period of time. But now, push those arrows way close together and you get a disappearance of the undulation. You go straight up but you reach a ceiling. You can't generate more than this maximum amount of tension, no matter how long you stimulate it. So you stay there until you release the tension and return. So this is called incomplete tetanus. This is called complete tetanus. This is more sustainable. This is the kind of thing that if you continue it long enough, you're going to begin to descend, to lose tension based on fatigue and cause essentially a cramping or a locking up of the muscle. Another way that this micro system supports long-term endurance or performance in an actual muscle is by the arrangement and activity of these muscle fibers into motor units. And this is a trick of the nervous system. One nerve, once it fires, is going to stimulate every fiber it's connected to. But within these large assemblages of fibers within fascicles and fascicles within muscles, you have the ability to connect multiple nerves within a single muscle. This is showing the biceps brachii, by the way, my favorite muscle, and showing you where we are illustrating the ends of these muscle fibers. Now you'll notice three colored groups, blue, red, and purple, that correspond to three neurons here in the motor region of the spinal cord. So in sending messages into that one muscle, the biceps, you're actually stimulating three groups of motor units. You stimulate the reds all together, the blues all together, and the purples all together. And if you time it, so you're alternating those stimulations. Two overlapping units are produce additive tension, while one is relaxing. And it looks something like this. So you're beginning by stimulating the red, and so the tension of the muscle as a whole only goes to this point because only red is contracting. However, you then begin, notice down here, you begin stimulating blue, so you're adding tension in the muscle as blue climbs. Notice you're peaking red right here, and you're already to this point with blue, and so the tension for the tendon, the end of the muscle, is all the way up here. It continues to build because even though red is dropping off, blue is peaking at the same time purple is starting. So there's a place here where you're actually getting some from purple, some tension from red, a high level of tension from red, and a high level of tension from blue. And that's what produces this high peak of tension at the tendon, where all these motor units are attached and transferring the power of their twitch. So by this sequential action and the motor units, we will be able to sustain what is actually a Treppe pattern. Look only at the red peaks. Notice that the red peak comes up here and peaks, but returns all the way to zero before peaking again. So even though these three motor units are generating tension up at this level, none of them actually get fatigued. So this is a highly sustainable action. We do see the delay of the stimulus. A small load can build a high tension quickly and drop off more slowly. Intermediate loads and large loads tend to compress. The distance that's shortened under a large load is less, and it takes less time. Now, we've been talking about a very high energy demand within muscles. We haven't asked the question, what's there to support it? Ultimately, it's all going to be ATP. But is ATP stored in muscles? Or are there other storage molecules that can be converted quickly to ATP when you call for high muscle exertion? And that's what this table discusses. In a standard resting muscle, there's some ATP, but very small amount, just enough for 10 twitches. 10 twitches will last about 2 seconds. So you basically, if you see a fight or flight situation, you decide, I got to run away as fast as I can, and you take off. The ATP there is gone in 2 seconds. But in a true emergency situation, a second molecule called creatine phosphate donates its phosphate to ADP as it's generated, making ATP and creatine. Basically, this is just a super quick transfer of phosphate from creatine phosphate to ADP, generating another 20 millimoles of ATP. It basically 70 more twitches, which basically takes you at maximum output for another 15 seconds. But it's very, very quickly depleted. And then we can rely on the third storage molecule, glycogen. Now, this is the same glucose polymer that's stored in the liver in the post-absorptive state. And in a truly rested body, these glycogen tanks in your muscles are full. Glycogen is stored directly in the muscle because when you have an immediate situation and you need muscular power, you need it quick. Now, it's a longer process occurring in mitochondria in the cells. Glycolysis can split the glucose very quickly. As we break this glycogen down to glucose, glycolysis splits quickly. 100 millimoles of glycogen in a typical fiber will give you 670 twitches based on glycolysis alone, and 130 seconds. This is very rapid, over 2 minutes. So you're approaching 3 minutes of exertion. 2.5 minutes of exertion, and you haven't really required any oxygen at all. But by now, your heart's beating faster, your respiratory system's delivering oxygen at a much higher rate, and this aerobic metabolism kicks in, and you have a number 12,000 twitches, or a sustained activity of 40 minutes based on the glycogen that's in your muscle. So it's important to understand the layering of energy molecules, and this output that we call the glycogen output is going to be supplemented by the transport of glucose and other carbon fragments into the cells by this increased circulation to your muscles. So we do have a storage capacity, but understand this portion of it that is the most extended exertion is just up to the aerobic maximum. It is limited by your ability to deliver oxygen. And the maximum oxygen transport that's possible when you're breathing as efficiently as you can and pumping blood as quickly as you can will set that aerobic limit. And that is where your exertion will stop accelerating. So let's take a look at 3 different states of the body. How is it that the body manages its nutrients in sustaining our muscular activity? Let's look first at rest, what we are now. The condition where the nurse would want to measure your temperature and take your blood pressure. And it turns out that at rest, we are recharging our muscles. So the blood vessels that are supplying oxygen and glucose and fatty acids by blood flow are interacting with the muscle fiber. The glucose is stored as glycogen until the glycogen tanks are full in the muscle. Other glucose is being stored in the liver, and we are managing glucose level in a relatively moderate range. Now, fatty acids are circulating freely, and generally beta oxidation is yielding those acetyl groups in high quantities and supporting all of the aerobic respiration that's necessary in the mitochondria of the muscles. This is why our breathing rate is so low. This is why our heart rate at rest is so low, is that fatty acids are sustaining this. And we are burning oxygen, we are generating CO2, and we are generating ATP. So at rest, what are we doing? We're storing up, filling our tanks with glycogen. If there is any bare creatine from a previous exertion, we are using ATP to phosphorylate it because ATP is surplus at this point. ATP forms ADP. ADP just goes back here and gets phosphorylated again. When the creatine is completely phosphorylated up to its tank limit, then this process slows and stops. And this is what I would call ongoing operation based on fatty acids, more than adequate to support our base metabolism, and recharging our tanks. Now, what happens when we begin exercising? Contract and extend these muscles. At this level of moderate activity, I'm going to define that as anything over at rest. So your heart does accelerate a little, your breathing accelerates, and it goes all the way up to the maximum yield of oxygen. So fatty acids are still there as a baseline, and they do form a rich supply of acetyl groups. So as long as you can beta oxidize and supply those acetyl groups, they're being fed in here, and oxygen is being consumed. Carbon dioxide is being generated, and aerobic respiration is taking advantage of a second source of acetyl groups that's shown here. As activity gets more and more strenuous and our breathing gets harder and harder, glycogen is converted back to glucose. This step is glycolysis. Now, its advantage, it requires no oxygen. But its disadvantage is out of the glucose splitting, you only get to ATP. So as your exertion calls for more and more energy, you kind of are backing up. You can only convert this pyruvate to CO2 so quickly. And pyruvate builds up. When it builds up, it's converted to lactic acid, and you get a soreness or a strenuous level of exertion. However, if you can complete this step, you're producing 34 more ATPs for every pyruvate you break all the way down to CO2 and water. So if you're somebody who trains for ultimate exertion events, maybe it's the biathlon in the Olympics, maybe you're a recreational runner, but you like to run marathons, or a recreational athlete who likes to do triathlons. These require strength, conditioning, and endurance. And typically, what conditioning means is recognizing and increasing and recognizing your maximum aerobic capacity. And in this picture, what we're seeing is breakdown of carbon fragments, but basically using the glycogen in the muscle as a source. Now, above that maximum activity, I was surprised when I ran my marathons, because I ran 26 miles, which was a long run for me. And I got into the stadium. I was tired, I was sore, I was breathing hard, I was fatigued. And I was surprised as the crowd began to cheer you on, you get a little excited. You actually have the ability to take off and do a short sprint. This is the anaerobic capacity that's on top of your aerobic capacity. So the stored high energy intermediates, the ones that do not require oxygen, kick in and add a little burst. Now it turns out you get excited and you take off running with 200 yards to go to the finish and find out you only have 50 yards worth of burst. But on top of the fatty acid and carbohydrate conversion we saw in the previous slide, we're going to add an anaerobic cap. So here's that pyruvate. This is now being diagrammed in the same way as the previous slide, but what we're really dealing with here is that excess glycolysis. The pyruvate goes to lactic acid, and lactic acid generates hydrogen ion that irritates the muscle and it makes you sore. It is, however, a source of additional ATP because it requires no oxygen. Over here, the creatine phosphate that we made at rest is still creatine phosphate. So when you do that stimulation, we call it sympathetic stimulation, that fight or flight maximum exertion. This is when the creatine phosphate gives up its phosphate and converts some of this ADP to ATP to sustain that burst of activity. So you can take off sprinting at the end of the marathon, and it lasts a while, but once the creatine phosphate is gone, you saw in the initial slide, that table, that creatine phosphate is only going to last about 15 seconds anyway, under the best conditions. At the end of a marathon, some of it has already been converted. But this is that balance of additional anaerobic mechanisms. It says most of the ATP, 2/3, is produced through this glycolysis. And when pyruvate hangs around, our body converts it to lactate. It's 1/3 of it is converted very quickly, a matter of seconds, into creatine, and the ATP that's extra stops. Now, after something like a marathon, you basically have this excess pyruvate because you've kind of gone aerobic at various places in your body. And that buildup of acid will produce an irritation and soreness. And for my marathons, I was a little sore the first day after. The soreness reached its peak. I really kind of got tender on the second day. But this lactate is kind of hard to clear. The body can come in here and break it down and transport it out of the muscle, and that's where you start to regain your flexibility and lose that stiff and sore feeling after a long event. We do have different types of muscles called slow twitch and fast twitch fibers that have different properties of endurance or speed. As you can imagine, slow fibers, you can see them here. They're smaller diameter, they're darker in color due to their own oxygen binding molecule called myoglobin. Hemoglobin for the blood, myoglobin can actually store some oxygen in the muscle and increase your endurance. They resist fatigue because of that extra oxygen supply. Fast fibers are larger diameter. They're very pale. They don't have myoglobin, but they contract very, very quickly. So they are easily fatigued, but they do produce very quick movements. You see the example muscle over here, a muscle of the eye movement. We move our eyes very quickly as we direct them toward what we want to look at. And that movement is moving a relatively light load. And so fast twitch fibers are found almost exclusively on those lateral eye muscles. So comparison of the properties, we do recognize intermediate fibers. The time to peak tension is prolonged in slow fibers. It's medium and intermediate fibers, very quick and fast fibers. We do see these different properties that extend to other systems. So high myoglobin in slow fibers was mentioned. It's low in intermediate and fast. Capillary supply, dense capillaries in slow fibers. So basically, the blood supply is going to go to the slow fibers, where you have the ability to process and use the oxygen and endure, whereas intermediate fibers have intermediate capillaries and fast fibers don't really have much of a blood supply. Many mitochondria, intermediate and few, as we move from slow to intermediate to fast fibers, kind of supports this idea of a slow fiber. Everything about slow fibers says oxygen supply, aerobic respiration, endurance, maximum yield, and maximum efficiency. Glycolysis gives you 2 ATP. Aerobic breakdown gives you 34 more. Everything about fast fibers says anaerobic, quick burst. Survival, 10 seconds may determine whether you live or die. Full speed at any cost. Intermediate fibers is kind of a blend of those properties that we find at various muscles in our body. Now, we've spent the whole chapter so far talking only about skeletal muscles. And I want to just mention that the other two kinds of muscles, cardiac and smooth, are appropriate for ANP2, where we deal with the cardiac organ, the heart, in the first organ system of the semester. And the smooth muscle is found throughout our other organs, the respiratory system, the digestive system, the urinary system, the reproductive system, all have smooth muscles. But so what I'm going to do for the rest of this chapter is just introduce a few basic differences about these muscles and do the real discussion of their function second semester in ANP2. So here we have cardiac muscle. You're doing something very different with cardiac muscle. By the way, it does form sarcomeres and it does have striations. Do you see these light stripes? But it's different. Cardiac muscles, or skeletal muscles, we said could be 12 inches long, and we're basically connecting them in linear arrays between points. So the biceps connects between here and here. They're long and they're fibrous. And what happens between those fibers isn't that important. But you're doing something very different here. You're making a bag and you're going to squeeze down on liquid blood, creating pressure. If this wall isn't strong in all dimensions, it just squirts out. So these cells are going to be shorter. They're going to be branched, an odd shape, and they're going to have these structures that bind them together with more force and that also support faster conduction of the electrical impulse across the heart wall. These are called intercalated discs. The muscle is, as I said, shorter. It typically contains one nucleus, in contrast to the muscle fiber, which contains many, many nuclei. And it basically forms a network of cells that looks like this. We do see striations because we still do have sarcomeres. Look at that sarcomere right there. But you also see this branching of the cell structure. So imagine a cell that comes out and forms two branches. Now you can basically interweave these branches with the branches of other cells, sort of like, it's not as regular as knitting, but it's the same thing, the idea. You're structurally kind of winding the cells around each other's at their junctions. Here you see one picture of the intercalated disc, and here another one. That intercalated disc is a special thickening of the connective tissue, forming little mounds that kind of basically intercalate with the mounds on the other side. More surface area means stronger connections. As a result, you also get a connection that conducts electricity much better. That's important to a pump. What do you want to do with a pump? You want to contract the muscle and eject the blood. Then you want to relax the muscle and fill. If you don't do that in synchrony, you don't have a good pump. If all the muscles don't contract at the same point, you don't get maximum pressure and you get an extended contraction cycle. If they don't all relax, then there's some resistance to the filling and the heart doesn't fill efficiently. So the cardiac muscle supports that function as a pump. As a result, there's a kind of a reorganization. All the parts are here. The sarcoplasmic reticulum, you can see, is needed to unlock the actin-myosin cross bridge. But you notice it has a different appearance. These T-tubules are short and basically still wrap this triad region. Short, broad T-tubules and a sarcoplasmic reticulum that kind of lacks those big pockets that we call the terminal cisternae. What we do see within the myofibrils of the cardiac muscle is a very dense array and regular packing of mitochondria. This is appropriate as well. Our skeletal muscles work hard when we call on them, but at night, they're happy to lie there relaxed all night and just recharge. The heart does not have that luxury. The heart begins beating when you're separated from your mother, and continues even at rest. Even your deepest sleep is accompanied by a steady heartbeat that's circulating your blood at all times. And so as a result, the constant supply of ATP is enhanced by this increased content of mitochondria. Smooth muscle looks kind of nondescript, but it's a spindle shape. A spindle shape would be like a kind of an oblong with points on the ends, an oval with points. Spindles were commonly known from spinning wheels, but we don't name the spindle shape that often anymore. So it may be a new term. You can take these cells and lay them down longitudinally. So the long axis here is left to right. And when cells run this way and you contract them, it would squeeze the tissue. It basically would contract the tissue from left to right, making this photograph thinner in this level, marked L. But up above you see a different picture. Here you're looking at a circular end, sort of like the pointed ends. And that's because the muscles are laid down in the circumference of this organ. So circling the tube, running basically straight at you out of the picture. So when this level contracts, it basically contracts in the up and down direction, squeezing the tube. So imagine I have a tube here in front of me. If I contract the circular muscle layer, it's going to squeeze that portion of the tube to a smaller aperture, close it down. And as it does that, it basically also pushes the tissue on either side away. So it lengthens that region. If I contract out of the tube and I contract the longitudinal muscle layer, it's going to shorten the tube. As it shortens it, it often basically expands the diameter that's inside. So as a result, this is a terrific muscle cell for managing tubes, managing bags. And we're going to find it. The cardiac muscle is special to the bag that's our pump that we call the heart, but all the other movement of muscles or control of movement. So for example, how do you change your circulation to send your the blood to your muscles, lungs, and heart during times of ultimate physical exertion? You do that by using the sphincter muscle, a smooth muscle. So the blood vessels that supply blood to the stomach, you don't need to worry about digestion when you're trying to save your life by fight or flight. So basically you contract a sphincter muscle and you close off circulation to the digestive tract. That increases the blood pressure in the body, and dilating the sphincters, the pressure opens up the blood vessels leaving the muscles, heart, and lungs, and all of a sudden you have more muscular performance. The orientation of these elements, now they are still actin and myosin fibers, but they're not organized into sarcomeres as we saw before, so they have no striations. Because we're not going to contract this muscle along the lengthwise axis, we're going to contract it in different directions. And this kind of a plaid appearance or diamond-shaped appearance shows the orientation of actin, myosin, and intermediate fibers around these dense bodies. And so when you bind the cells together and contract them, you're basically going to shorten them left to right, but you can see they're also pulling them in at these diagonals, top left to bottom right and top right to bottom left. So the idea in shrinking a wall is to shrink it in all dimensions, not just its length. We will return to smooth muscle in detail on this fascinating system later. So when we compare these, we see different properties of dimensions and nuclei and control of calcium in producing the single thing that all muscles share, which is contraction, movement. Skeletal. Skeletal says fibers pulling straight and then being able to fully rest. So typically skeletal muscles cross joints. If they are completely in soft tissue, they produce a movement of soft tissue like the lips or the eye openings. Cardiac muscles, a muscular bag that's contracting and relaxing, and this is an important word, constantly. Never stopping the heart is a bad thing. Always pushing that liquid forward and maintaining a positive blood pressure. Always, always. Stop the heart, you drop on the floor unconscious. Smooth muscle. These are contraction in layers. They're going to form sheets. They're going to form sphincters throughout our organ systems. It's going to be able to produce contractions that move along the surface of an organ in waves or in ripples, or produce a particular line of contraction in the tube. We're going to manage our tissue walls and move them in many ways based on smooth muscle. So, good news. We got through muscle tissue in count them, 2 sessions. And that means we will be able to post the entire chapter by tomorrow.